Welcome if you are joining us for our Quarantine Science Cafe. For those of you just joining, if you could find your chat box and if you could just introduce yourselves, let us know who you are and where you are from. We're going to give a couple of minutes just to let people get on in today. So when you introduce yourselves, make sure you choose to all panelists and attendees so everyone's gonna see who's here, where you're from. It's nice to see all of you who are already typing in. I see some familiar names and some new ones. I see someone from the great and powerful Rhode Island. I might dispute that with you, having lived in Rhode Island for a little while myself. See, we have some Mainers. I see we have our friends from New Mexico here today. That's fantastic. Looks like New Hampshire, Vermont, New York. I love that we are really getting a nice wide range of folks from all over the country. This is fantastic. Just going to give it a few more minutes. I'm watching how many attendees we have in and knowing how many we're expecting. Just going to give it another moment. So keep introducing yourselves. Thank you all for doing this. I'll introduce myself and our presenter once we get started. Love seeing where everyone's from. Hope everybody is having a good Wednesday. It is Wednesday, right? I've seen the comments that you have to name the days now. This day, that day, the other day, someday. Has anyone seen those comments? We got some more Mainers. We have someone that's from Vermont and recently back from living in France. That's fantastic. So I am going to get us started today. My name is Lauren Traster, and I am the 4-H Teen and Leadership Program Coordinator. And we put on these quarantine virtual science cafes each and every Wednesday. So thank you for joining us today. Just want to go over some of the protocols we have for our virtual gatherings. So first and foremost, you all are muted today. You don't have the ability to speak out loud, but you will be able to share your thoughts and your questions in two different ways. The first is our chat box. You can share any comments or thoughts about today's presentation in the chat box, but we do ask that you limit really side conversations or anything that's not on topic. We don't want the chat box to end up being distracting. If you find that there is a lot of chat going on, you can always close it so that it isn't distracting to you, but please just be considerate about what you post in the chat box. Please do not put questions there. If you look at your menu bar, if you hover your mouse towards the bottom of your screen, you'll see that there is a Q&A box. That is where all questions for our presenter will go, because we'll be able to see the questions there. And actually, if you go into the questions, if you like a question someone asked, or someone asked a question you were planning on asking, you can actually, there'll be a thumbs up there. You can click on that. And what that does is it lets us know that more than one person has that question, and actually moves the question up into a higher ranking so that we make sure to get that. So we're going to ask that you're courteous and respectful today. Again, what you write in the chat box, what you write in the Q&A, please just be courteous and respectful to our presenter as well as to one another. I've mentioned this already, just please do not create any distractions. We're here to learn, and if you're not here to learn, you can choose to leave at any time, but I know our presenter is really great. We've had her present in the past in person, so I know all of you will learn a lot today. So I hope that you stay engaged and participate fully in our cafe today. So I want to take the time now to introduce our presenter, Dr. Heidi Malaby. And Heidi is a grant proposal specialist working at the University of Vermont, assisting faculty, doctors, and professors campus-wide from the most innovative ideas to fix the world's problems. She recently completed a postdoctoral fellowship at the UVM Larner College of Medicine, where her research focused on how special proteins align chromosomes during cell divisions, a process that is disrupted in cancer cells and is therefore targeted for cancer therapies. Her research used really cool microscopes to image cells in the process of dividing. Dr. Malaby completed her PhD at the University of Massachusetts Medical School in Worcester, Massachusetts in 2014. So I'm going to stop sharing my screen and let's welcome Dr. Heidi Malaby. And Heidi, you can just hit the share screen to share your presentation and get going.
All right. Yep. So hi, everyone. Thank you for joining us today. So let me go ahead and pull up my presentation here. All right. And thank you, Lauren, for that great introduction. So as she mentioned, so I'm not currently working in the lab, but I just finished a postdoc last fall. And during that time in the lab I was working in, one of the research projects I did was to create a human CRISPR cell line to study a specific question in the lab that we were interested in asking. So I'm going to share that story with you guys today. So I wanted to start by taking a quick poll and see how familiar you all are with CRISPR. So these are the choices you have. You have, I've never heard of it, something about designer babies. I know it's a genetic tool that can change the sequence of DNA in live cells, or I understand what clustered regularly interspaced short palindromic repeats means.
So I just launched the poll. Hopefully you all see it. And if you don't, you can put the answers in the chat box. But I see people taking the poll, so I'm guessing you see it, which is great. So how familiar are you with CRISPR? Never heard of it? Something about designer babies? I know it's a genetic tool that can change the sequence of DNA in live cells. I understand what clustered, regularly interspaced, short palindromic repeats means.
All right, great guys. So this looks like probably just about everyone. So it looks like 57% of you know that it's a genetic tool that can change the sequence of DNA in live cells, which is awesome. And you're at the right talk to hear a little more about that today. And then our next group, we got 40% that say they've never heard of it. So you guys are in the right spot too, because you're going to hear about it today. And I see no one understands what clustered regularly interspaced short palindromic repeats means. And that's okay, because honestly, I don't really know what that means either. And that's not the point of using CRISPR as a tool. So we're actually not even going to talk about that today. All right, so we can close the poll. I'm just going to close out of that. All right, so as for those of you who have heard of CRISPR, you probably have heard about it.
Heidi, can I just interrupt?
Yeah,
we see your full desktop. Can you put your slides in the slideshow view?
let's see. Oh, I have to resume the share maybe? Now, is that better?
Oh, there you go. Yeah.
Okay, it paused it when we went to the poll, I guess. Okay, that's good. All right, that makes sense why I couldn't go forward in the slides too, I think.
Yeah, so now we see the poll slide. So you might want to advance one more. There you go.
There we go. All right. Okay, so the for those of you who have heard of CRISPR, you may have heard about it in the news because it is kind of a buzzword right now in the medical community. And that's because it has the potential to revolutionize the treatment of medicine. And I'm not going to talk about this too much because I want to focus on the actual technique itself. But just a month ago, on March 4th, the US started its first clinical trial using CRISPR to edit underlying gene mutations in a disease that is causing blindness in people. So this is really exciting, right? This has never been possible before in medicine to actually cure someone by treating and fixing the underlying genetic mutations. So because of this potential, we hear about it a lot in the news, and it gets a lot of attention. The other flip side to this coin is that there's a lot of fear around the ethics of this technique, right? So if you've heard about CRISPR babies or if you've heard about designer babies, in theory, that is possible with the CRISPR tool. That if we know what genes to change in a certain way in a person, we could in theory design, quote, an ideal human being. So there's a lot of fear around this technique as well. And so this is an article from last year because someone in China, there was a researcher in China who actually did this with human embryos, trying to make HIV resistant children. And first of all, he did it very badly. If you've heard about this story, someone who's familiar with the technique, he did not do it very well, and those children are not going to be HIV resistant to begin with. And two, the ethics around changing a new generation is very concerning for a lot of people. So I'm not going to talk about this level of CRISPR anymore, but if you're interested in understanding this idea, especially about designer babies, if you have not heard of the movie Gattaca, I think it's from the 90s maybe. It's before CRISPR was even discovered, but it is basically a science fiction movie about CRISPR. So go ahead and check that out if you're interested. All right, but what we're here today is we're going to focus on CRISPR as a tool, and we're going to focus on the molecular level of how do you actually make a CRISPR cell line. So I am actually going to start by showing you a video on YouTube. So I think in order to do this, I need to stop sharing my screen for a minute and jump over to my Chrome so you can see. All right, Lauren, can you see this screen?
Yes, and so I think you're gonna wanna probably hit the expand button in YouTube to fill your screen. Yeah.
All right, so this is just a couple minutes, and it's gonna talk about, on the molecular level, how CRISPR works.
Every cell in our body contains a copy of our genome, over 20.
And Heidi, all we see is a gray screen.
Oh, really?
Yeah.
Okay.
Consists
Can you
of
hear
two
it?
strands
We can hear it.
helix
Okay.
and held together by a simple.
I'm gonna go ahead and have you listen to it then, even if it's just grainy.
Oh, actually, now we see. I think there was just a delay.
Okay, that may have. Let me try to exit the full screen. Let me play a second of it from here and see. This might be a little better.
Okay. Yeah.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of two strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use two tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut, but the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function.
All right, so I'm actually even just going to stop it there because that's all that I'm going to talk to you guys about today. I apologize if that was grainy, but I hope you could at least hear it. And this is on YouTube. It's made by the McGovern Institute, so you guys can go and watch it later if you want to see a more high quality image. But so that is basically the overview of what's happening on a molecular level with CRISPR. And so, you know, the project I was working on in the lab was to do essentially what's shown in the screenshot, which is to target a gene that we were studying in a very specific place using an enzyme called Cas9 that was directed to that spot by a small RNA called a guide RNA. And using that to repeatedly doing this in a population of cells to eventually find a cell that is not repairing the DNA properly, which is taking out the gene, so it's not able to make the protein we're studying. All right, so I'm going to go over that in respect to the actual project I was doing in more detail. So I'm going to stop sharing this, and I'm going to go back to my other presentation. I'm actually going to close out of Chrome so my computer is not as angry. And then I need to share this again. Hold on. Share screen. PowerPoint. All right, is everyone back in PowerPoint now? Lauren?
You're back in there. It's just, I think there is a delay on showing the actual screen because it's just white at the moment.
Oh, it's paused again.
Maybe like, yeah, there you go. Okay, now we got it.
The pause thing again. All right, next slide. All right, so why did I want to make a CRISPR cell line, right? So what were we actually studying in the lab? So the lab I was working in, we were actually studying cell division or mitosis. So on this slide, you're seeing images that we actually took in the lab of cells where we've labeled different components of the cell to show the whole cell cycle, right? So I'm not going to go through each of these phases on their own. These are very classically known cell cycle phases that we've known for over 100 years. But what's interesting is we still don't really understand a lot of how this process is working. So the phase of the cell cycle that I started studying was metaphase. And then I'm going to talk to you about not just metaphase and our understanding of how chromosomes are aligning nicely on the metaphase plate, but also how the chromosomes are dividing to each end of the mitotic spindle, right? This is the mitotic spindle in red. And how 2 new daughter cells are formed from one starting individual cell. All right, so mitosis, you know, if you have studied mitosis in school, you've probably seen pictures like this, maybe cartoons, maybe not quite as colorful and cool as this, but there you see static images, right? But cell division is not a static process at all. It's actually very dynamic. So the reason that this process is dynamic is because the mitotic spindle, which is the red components here, is actually made up of a protein complex called microtubules. And the microtubules are actually formed by these small protein subunits. So these little like orange and green subunits. This is called a tubulin protein. And these guys polymerize into these long hollow tubes, thus microtubule, right? It's a small tube in the cell. And they go through periods of both growth and shortening. And if we actually could take one of these microtubules out of the spindle and look at it in isolation, which we did, you can actually see the microtubule growing and shortening. So this is an actual microtubule on a slide that is going through these periods of growth and rapid shortening. So if all, right, and so this is just one microtubule, and in a spindle, there are thousands of these things. And they're all connected at the end that grows and shortens. They're all connected to chromosomes. And so if this happened in an uncoordinated manner, chromosomes would just be all over the place all the time. It'd be mass chaos and it would never work. So what actually happens is that they coordinate and they go through periods of growing and shortening at different times in order to align chromosomes, right? So here's another movie. So now we're looking at the chromosomes in green, and we don't see the spindle, right? So we don't have that labeled in this video, but we do have labeled kinetochores, which are the protein complexes on chromosomes that the spindle is connecting into, all right? And that's not so important for this video, but we're gonna talk about this more later. But so here, focus on the chromosome. So this is what it actually looks like in a cell as chromosomes are aligning, right? So this is leading into metaphase. See, they're actually moving back and forth, all of them quite a bit, until they align, and then they divide to each end of the cell once they've all satisfied that alignment characteristic. All right, so this whole process, there's a lot of regulation and there's a lot of dynamic movements that are happening to get to that chromosome alignment period and getting the chromosomes, each copy to each end of the cell. All right, so the way that regulation happens is there's a whole bunch of different proteins that are in turn regulating those microtubules, which are in turn pulling and tugging on the chromosomes, all right? And this now, I've gotten you to the level, this is what I was actually studying in the lab, is this particular family of proteins called kinesin motor proteins. And so this is a little animated video, and this is the kinesin right here, and it's walking along this microtubule down here. And this is it's a dynamic video, it's a molecular simulation video of what these proteins actually do in cells. So they're actually little proteins in your body. There are hundreds of these in every cell that actually literally walk along like this on this little subatomic track of a microtubule. And so there's a lot of these that have different functions in cell division. And I was studying one particular one that has an absolutely terrible name. It's called KIF18A. It stands for kinesin family, something like that. And it's like member 18, number A. Like it's a terrible, terrible naming system, but a lot of biology is like that, unfortunately. But this particular protein, though the name is forgettable, does do something very interesting in cell division. And so this was the protein that we wanted to to disrupt the gene using CRISPR to create a cell line that would not express KIF18A. And the reason we wanted to do that is because we already knew that KIF18A does something really, like when KIF18A is not in cells that are dividing, it does something really interesting. And we know that because there are other techniques that we can use to remove the protein for a really short period of time, and look at cells that don't have it for a short period of time to see what's happening. All right, so when we did that with cells, what we found was that the chromosomes didn't actually align, right? So this is an example of a cell in metaphase, and instead of having all the chromosomes, which are now in blue, line up in the metaphase in the middle of the cell, instead what we see is that the chromosomes are kind of spread all over the place. But they're still attached into the microtubules, right? So this situation is unique. You don't usually see this when you knock out a protein in cell division, and even more so that even though the chromosomes were unaligned, right? So this is a video now that we're looking at just these protein complexes of the kinetochores. You can kind of see the outline of the chromosomes in the cell as well. But you can see how they're not aligned at all, right? They're all over the cell. But even though they're not aligned, the cell is still able to go through cell division, right? So this finding was like paradigm changing, right? Like we've known for 100 years that chromosomes align in metaphase before they divide. Like that's what cell division is you align your chromosomes and then you divide them into two daughter cells. So here is an example now where the cells are not aligning, but they're still able to separate and go out into each daughter cell. So this then is the question that we wanted to study, is if we could make a cell line without KIF18A where the cells are never aligning their chromosomes, what are the consequences of this, right? What are the long-term consequences of having mitosis with unaligned chromosomes? And this question could only be answered by removing the actual KIF18A, the functionality of the KIF18A gene from cells. All right, so now jumping back to how did I actually make a CRISPR cell line? All right, so these are screenshots, perhaps clearer than from that other video that I showed, with some labels more specific to what I was doing, right? So I'm not gonna take the time to go through how I actually designed all of these components, but the gist of what I did, right, was I introduced Cas9 enzyme to target the KIF18A gene in this human cell line that I was working with. And then I designed a guide RNA piece, which is here in red, that would match up with a particular region of the KIF18A gene. And this targeted the Cas9 enzyme. And then the Cas9 enzyme creates a double strand break in the KIF18A gene. And then when this happens in enough cells over time, eventually, right, so normally when there's a double strand break in the cells, in the DNA of a cell, the cell will repair it, right? There's all types of regulatory mechanisms to ensure that our genes like don't get messed up, right? Because we want to live for a while with maintaining our genetic integrity from cell to cell. But no one's perfect, including our repair mechanisms in our cells. And at some rate, the cells will eventually screw up the repair process and they'll introduce a mutation. All right, so the goal we were looking for here was to have some type of frame shift mutation that would prevent the cells from being able to, create some type of mutation so that they would not produce a viable KIF18A protein. Lauren, do you want to say something?
I do. So there's a lot of questions that have come in that I think might be helpful to have the answers as you continue to move on.
Yeah, please.
Okay, so, and a lot of these have been upvoted. So a lot of participants want to know, are these real images or computer generated?
The images of cells are real. So the movies of the cells I've shown you, those are all real. There was a cartoon of a kinesin protein walking. That is a cartoon that's made up. We cannot visualize proteins on that magnification. So that is someone's artistic rendition based on scientific data. These images we're looking at right now, this is another molecular simulation, right? Because we're looking at, these are anything that's trying to show you an actual protein. We don't have that type of resolution yet.
A lot of participants want to know, how much do the microscopes you use cost?
Hundreds of thousands of dollars is the main one we used. So, and it was a fluorescence inverted Nikon microscope.
Was the microtubule animation a time lapse or the actual speed?
That was a time lapse. Yep. So how much of a time lapse was that? That was probably sped up.
I thought it said 200. I thought it said 250 times on your slide.
Okay. I think I don't, honestly, I'd have to go back and look. I don't want to do that right now. But yeah, so the microtubules, they'll build up tubulin dimers on a matter of minutes. And then the part where the microtubule shrinks, that'll actually go, that will happen very quickly. That'll happen in a matter of seconds. Cell division itself, the process of aligning chromosomes and in metaphase, that takes 20 to 30 minutes usually, depending on the cell type, but 20 to 30 minutes is pretty standard. And then anaphase is pretty fast. That usually happens in maybe a minute.
Great. Another question, could CRISPR potentially assist gender affirmative care in surgeries?
Let's save that one for the end.
Okay. We'll come back to that.
That's a great question, but it's a little tangential to the story right now.
Okay. Is that a GIF animation of serotonin?
No. Yes, so you're referring to the kinesin motor protein animation. That's right. So yes, so there was a GIF that went around a number of years ago that supposedly was a kinesin motor protein transporting serotonin, and it was something about like you're literally seeing happiness or something like that. Unfortunately, that was not an accurate GIF, and I'm very sad about that. My mom sent me that too, and was like, "Is this real?" So there are other motor proteins called myosins, and myosins are the ones that transport serotonin along neurons. So there are lots of different types of motor proteins. Myosins look differently than kinesins though, so that's why you could tell that the GIF was not accurately represented there. That's probably enough of an answer for that question right now.
Great. Well, I'll let you know when there's a lot more.
Okay, great. All right, so let's jump in. Okay, so this is the object of what I was trying to do with this project. So how did I actually do it? All right, so we're gonna go over the actual process I used in the lab to create a CRISPR cell line. All right, so the first thing I had to do was to add the CRISPR materials into the cells in culture. And for this project, we wanted to use a human cell line, and the cell line we were actually using is the one that I've shown you in the videos up to this point, and they're actually from a human eye. So they're called human epithelial retinal cells. All right, so the first thing is we have to get those CRISPR materials into the cells so that CRISPR can happen. The second step is we have to identify which cells has CRISPR happening in it. And then we have to be able to isolate those cells, grow those cells up into their own new cell lines, and then find a way to identify which of these cell lines actually have the KIF18A gene mutations that we want, which in this case was just trying to knock out the gene so it's not functioning correctly. All right, so we're gonna go through each of these steps individually. So first, how did I get the CRISPR materials into cells in the first place? So I used a technique called plasmid transvection. This is a very common technique in cell biology. And so it involves adding the DNA for different, that would code for different genes into cells, right? So I had and I should say a plasmid is a circular piece of DNA. And this is actually, it's also known as a bacterial chromosome because bacteria, instead of having chromosomes like the ones I just showed you a bunch of pictures of, the bacteria just have one circular piece of DNA that is called a plasmid. All right, so we have found ways to engineer these because they're pretty small. So anything that's molecular biology based, it is very often you're working with plasmids. Right, and so what we did was we used plasmids that had the gene for Cas9 enzyme on it, and a plasmid for the guide RNA gene, which would then be transcribed into RNA. We had these on plasmids, which we then fed into cells. And the other component of the plasmid that I wanted to mention that's important is that we didn't just add a plasmid with Cas9, we added Cas9 tagged with the GFP gene. And so GFP, if you guys haven't heard of it, stands for green fluorescence protein. And so this is, on the plasmid, it's the gene, but it codes for a protein, which this is a cartoon down here of what GFP looks like. And this is a really special protein because when you shine blue light at this protein, it actually emits green light. And this is a protein that's natively found in deep sea animals like jellyfish. And they actually use this protein to attract different prey in the deep sea where there isn't a lot of light. But so someone discovered this and had the brilliant idea of using this as a tool to image things in cells. And also as an aside, a lot of those really cool pictures I was showing you at the beginning that we imaged, those were actually cells that were expressing different proteins that were tagged with GFP or a different color of protein of the similar family. All right, so we have our materials, we have them on a plasmid, and then to get them into the cells, we have to coat them in lipids. And this is because this plasmid by itself has a hydrophilic surface, meaning that it won't get past a plasma membrane without having a lipid coating around it, right? So this lipid coating creates a hydrophobic surface on the plasmid, and it allows it to get inside of a cell past the plasma membrane, right? And then once it's in a cell, then our cell machinery will recognize that this is a gene and that is gonna code for a protein. So RNA polymerase and the nucleus will actually just register that, oh, this is something I can make into mRNA. And then this in turn will be translated into a protein. All right, so I didn't put the central dogma of biology on this slide, right? That DNA codes for RNA, which codes for protein. If you guys don't know that, that's a thing, right? So that's usually the standard of how we get from genetic code to an actual protein that is actually doing something in the cell, right? Okay, so this is how we started. So to get the CRISPR materials into this human cell line, we did a plasmid transfection to get them in the cells. And then once Cas9 was translated, it would light up green because of this GFP tag. All right, so the next step was, oh, this is slightly out of order. Anyway, so the next step was to actually, in that population of cells that we had treated with the CRISPR, with the Cas9 and guide RNA plasmids, how were we going to identify which cells actually received those plasmids? So to do that, I used a technique in a machine called fluorescence activated cell sorting. And so this is a picture, and this is a picture I took of cells in culture that were treated with a blue light, and you can see that some of them are lighting up green, right? So these are all cells that are expressing this Cas9 protein in the population. But you can see also that there's a bunch of cells in here that are dark, right? They're not lighting up, right? So, and in a dish of cells, we're talking like 2 million cells in a dish, and only 40% of them actually have the Cas9 and guide RNA protein and RNA to begin with. So one of the first steps in getting a new CRISPR cell line is to just isolate the ones that have the material. All right, so this machine can actually sort out the green cells from the dark cells. And how it does that is with a laser beam. It shoots it, well, I should say, so it uses a very fine capillary to pass the cells one at a time, pass a laser beam on one side that has a detector on the other side. So what this does is, as the cells are coming through, they go through this light source, and if the green detector recognizes that they're expressing this GFP, then they're sorted into a GFP pool. And the ones that are dark will end up in their own pool. All right, so this is a machine that we can just grab the GFP expressing cells and nothing else. Now, because we're making a CRISPR cell line, this isn't good enough, right? Because the way that Cas9 is gonna be treating each of these cells is gonna be different, right? So a lot of the time, the cells are gonna be able to repair that KIF18A gene just fine, and only a small percentage of the time is it actually gonna mess up, right? So we actually want to look at each of these cells individually. So we took this one step farther, and we had this machine, instead of just pool a bunch of cells like this for us, we had it take individual cells and plate them one per well into this 96 well plate, right? And then when I did this experiment, every time I did this, I had like 4 to 8 of these 96 well plates worth of cells after this sorting step. All right, and so now we have green cells. We have one of them per well in these small plates and small wells. And then the next step is to grow these isolated cells up into new populations, right? We're making a bunch of new cell lines, basically. And so here we started at a 96 well plate with 1 cell per well, and we let these just sit and grow until they became dense in their little wells here. And then we moved them into slightly bigger wells. And then when they were pretty dense here, then we moved them again into these considerably bigger wells than when we started. And from this step to this step, I actually divided each of these new populations up into 2 different wells. One so that we could keep the cell line going in case it was the one we wanted. And then the other well we took to do the next step, which was to see if the cells actually had KIF18A knocked out. Lauren, did you want to say something?
No, we do have a bunch of questions, and
Yep.
just letting you know the time.
Okay, cool. All right, so then the next step was to actually screen and see if KIF18A was working, if KIF18A was knocked out in any of these new cell lines. And so this, I'm gonna go over this pretty quickly because this whole part could be its own talk. But we started by taking one of those wells and extracting the genome of the cells for each of these new cell lines. And this sounds complicated. This can actually be done in about an hour, maybe 2 hours, and you can do a bunch of them at once. So this is actually a pretty easy step. And then what we did was we put these cell genomes through a polymerase chain reaction to amplify the region of the KIF18A gene we were looking at. And this is complicated, and I'm not going to go into it with a bunch of detail beyond to say that this is a technique that you can start with one copy of the thing you really want to look at, and through a bunch of cycles, make a whole bunch of copies at the end of it, right? So we can't see just this by itself, but if you put it through this whole thing, which also, it goes pretty quick, in like an hour or two, you can get from this to thousands of copies, really. And this is a way to amplify and look at just this region of the KIF18A gene we wanted to look at. Now, the other thing to think about, right, so at this point, we could have just taken these and sent these samples for sequencing to see if the KIF18A gene was mutated. But we knew that most of the cells we were looking at were going to be wild, were going to be completely normal for the gene, right? And we knew going in that only about 5% of all of these new cell lines we were looking at were actually going to have mutations in the KIF18A gene. So I engineered, when I went through this whole design process, I engineered in this little piece of DNA a unique restriction enzyme cut site that, if the sequence was normal, would cut this piece of DNA into two. And if it was not normal, it would not cut. So what that looks like is this, right? So this is DNA that's been labeled with ethidium bromide and run on an agarose gel. And here are the first two lanes. These are just untreated cells. And down here you can see that in this case, there was no enzyme added, and here there was enzyme added. All right, so this is what like this piece of DNA looks like normally on this gel. And then when we add this restriction enzyme, this is cut into two different pieces. And then what I saw was that for each CRISPR cell line, also treated with the restriction enzyme, they would either cut or not cut, right? So I saw a lot of gels that looked like this, where the normal sequence was present and it was getting cut, right? So there were lots of things that look like this. And I don't remember how many cell lines I ended up screening, but it was well over 100. But eventually I got one and it showed me this, which was super exciting, right? So so here we have, this is the same untreated cell conditions where we have it's not cut, it's being cut. And then here, this CRISPR cell line, it has the enzyme, but it's not getting cut. So that meant that this cell line was mutated in that spot. And so this cell line I went and sequenced, and we were able to confirm that there was a frame shift mutation and KIF18A was knocked out of the gene. So it worked. So I was actually able to make a KIF18A CRISPR cell line. And then when we started looking at the cells, this is what we saw. So these are all actual cell images. These are all cell images that I took. And these are all cells in metaphase. And what I hope you can appreciate is that in this KIF18A knockout cell line that I made, the chromosomes are not aligned, just like in that earlier slide I showed you, right? So this is what it should look like normally in metaphase, and this is what this KIF18A knockout cell looked like. All right, and I'm gonna fly through these points because it's not as, this is all validated. Right? Basically, I did a bunch of controls to validate that we hit KIF18A gene only. And we're very confident that the cell line, this is what we're seeing when just KIF18A is removed from cells. All right, so then the last thing I want to finish with is to show you the result to answer that question. Like, what are the long-term consequences of cells dividing without aligning their chromosomes? So this is our second poll, where I want you guys to predict, what do you think happened in the cell line when we started looking at cell division? Do you think the cell's division occurred normally? Do you think that more chromosomes went into one daughter cell than another? Did the chromosomes divide equally, but the daughter cell nuclei looked weird, or all of the above?
And the poll is being launched, so you should probably see it any minute now.
All right, great. So it looks like we got a pretty even spread, which is awesome. And this is actually slightly a trick question, honestly. So you guys are kind of all right. So good job. The biggest things we found, which is the one that most of you selected, is that the chromosomes divided equally, but the daughter cell nuclei looked weird. But actually, this only happened in 10 to 20% of the cell divisions we looked at. And the rest of the time, the cell division occurred normally. So my bad on putting the all of the above on there. I actually should have done like A and C. But nice job for participating. Thanks, guys, for staying with me. And for those 15 of you who chose C, and the 8 of you who chose A, nice job. Okay. I'm going to go stop sharing, close out of that, and then do you see my screen, Lauren, or do you see my, okay, we're good. Okay, so that didn't happen that time. Okay, so this is what actually happened. So, so there were 2 weird things that we saw from this. The first was that sometimes during cell division, when the nuclei were reforming after cell division, there was micronuclei that appeared. So this is a movie. You can see, we're looking at the chromosomes, and you can see that, again, they're all over the cell. They're not being aligned at all. And this goes into anaphase, and I would want you to watch one of the trailing chromosomes. All right, so here's this chromosome, and you can actually see as the daughter nuclei are reforming that chromosome never makes it in with the rest of the nucleus. All right, so this is what's called a micronuclei in cells, right? So that was the first weird thing we saw, and we saw that in about 10% of the cells that we saw going through division. The other weird thing we saw was that there was some crazy lobed nuclei that were happening, right? So normally when you look at a nucleus in a cell, it's kind of this oval shape, but it's fairly well formed without any weird kinks or other abnormalities. But in this KIF18A knockout cell line, we saw a lot of nuclei with funky shapes, ranging all the way up to some like really crazy ones that don't look anything like this at all. And I'm going to show you a couple of videos of what that looks like. So this is a normal cell dividing, and here we're looking at, this is an overnight movie, so it's a longer time frame than what we were looking at in the last movie. But these are, again, we're looking at the chromosomes. So here you're seeing the nucleus. And when I play this, you can see that this cell is in interphase for a while, and then it goes into cell division, and it forms 2 daughter nuclei that both look pretty oval and normal. But then in this KIF18A knockout cell line, here, I got 2 examples of this just because these are so fun. Check out the nuclei in the very middle of the first image here. You see, it divides, and then look at all the crazy things that happen, right? So we got like 3 micronuclei down here, got like a massive one up there. Those are not normal cell divisions, right? And this one has like a crazy lobe off to the side. You can also see a bunch of cells down here that are already in interphase that have micronuclei floating around, right? That's weird. You don't normally see that in the population. And then in this example, again, watch the guy in the middle. You see it goes into cell division and divides, and insanity ensues. Look at that. Look at all those crazy little things that pop up. So that is not a normal thing that you see in cells happening. And you can even see in this video up here at the beginning, there's actually a cell division that occurs normally, right? So there are some cases where the cells can get by just fine with not aligning their chromosomes, but then other times we have these really crazy effects. All right, so I know we're running low on time. So there we go. So I'm gonna finish here. So the real reason why we need to align our chromosomes before dividing is to ensure that the chromosomes remain in order in the nucleus for the next generation of cells. Right, so this is not a healthy thing for cells to be doing in the next population. And there's always one more question why, as to why, because one thing you could ask is like, why is this even an issue? Why can't cells just deal with having slightly weird nuclear shapes? And to briefly answer that question, nuclei and chromosomes and nuclei kind of actually each have like their unique zip code where different regulatory processes that happen in interphase are looking for chromosomes in certain areas. And if those chromosomes are not in those locations, it's much harder to do those functions, right? And these are all different types of interphase functions that need to happen based on where these chromosomes are located. All right, so this is a really exciting story. I was really glad to be able to work on it. I'm gonna stop there and we can take some more questions. Just wanted to mention again that this is work I did at the University of Vermont, and I worked with Jason Stumpf and his lab, and these were the people that were in the lab at the time when I was doing the work. And this, all the work I talked to you about today is actually published and it's freely available. So this was published in February of last year at the Journal of Cell Biology. And so if you Googled this title, you could go and access this paper, and there's more like movies and photos and stuff of different cells that we imaged on there if you're interested. All right, so I think I'll stop there.
This is great, Heidi. Before we go to questions, I'm going to launch one final poll. I just would like to get some feedback because I know some of you probably will have to log off soon, but we will answer the questions. Just take this last poll, and this should get launched in a second. You're going to see on a scale of 1 to 5, please rate the overall cafe experience. And then on a scale of 1 to 5, please rate new knowledge gained. So did you learn something by coming today? And you can put any comments you want in the chat box if you want to tell us, like, you know, something specific you learned. But once the poll is done, we'll go right to the questions. So for those of you who ask questions or want to hear the questions, please stick around. And also, we do have a cafe next Wednesday. We'll send you out the information. Hopefully you'll all come back again. So thank you all for taking. Heidi, do you see the Q&A box?
Yes.
You can open that up and start with the question.
Okay. All right, so let's see. So the top question is, how do you remove the gene strand from the human eye? So let's see. So I guess that could be phrased two different ways. Let's say that's like the one I was talking with at the very beginning. That's the clinical trial that's happening now, right? So what is happening with that clinical trial, right? So, okay, so here's what I should say too. So the main drawback with CRISPR is that you have to, if you want to change the gene of a cell, and you want it to have an effect on a person, you have to do all of the cells that would in the body, basically, right? So like if I wanted to change my KIF18A gene in my body, I would have to do this on every single cell in my body because I'm already a grown adult, right? And I would have to target each cell individually in order to get that change, right? And that's one of the bigger drawbacks with CRISPR, using it as a technique, is whatever cells you want to change, you have to do it on like every single cell in the body. It's not like you can just take a pill and it'll magically change the DNA in every cell of your body, right? So the way that the clinical trial is working for that human eye blindness study is that it is introducing the Cas9 and a guide RNA against a specific gene that's mutated, that's causing the blindness, and it is doing it in all of the retina cells where that protein needs to be functioning correctly, right? So that's how that technique works in theory, and then we'll see if it actually works in clinical trial. All right, so I'll move on to the next question. Could CRISPR potentially assist gender affirmative care and surgeries? Right, good. So this is a good second question from this. So this is a great question. And the answer is similar to my answer for the last one, which is that you could do this, but you have to do it on every cell in your body to make changes on the gene, right? And the other big thing here, right, is like changing it, like every chromosome has hundreds to thousands of genes on it, right? So if you wanted to like change an X chromosome to a Y chromosome or a Y chromosome to an X chromosome, that's a lot of genes you would need to change. So that would be much harder to accomplish because of the scale of the number of genes you would need to hit. And then on top of that, you'd have to do that for like every cell in the body. And even if you just targeted reproductive organs, that would be really challenging to do. But I like the imagination. Will we have access to the slides? Lauren, what's the question for you? I mean, I can give you the slides if you want it to post them.
Yeah, I mean, if you're willing to provide them, if you want them, you can send me an email and I can send them out. The other thing is this is being recorded, so you can also have access to the recording, which will go back up on the website where you register.
Great. All right, is it possible to reuse the cells that didn't make it through the fluorescence detector? The answer is yes, you could definitely do that if you wanted to. As someone who did a lot of cell biology, I will say cells grow and divide very quickly. And the issue is not the number of cells, but the resources to maintain the cells, right? So cells in culture divide about once a day. So every 2 or 3 days, if you have cells growing in a dish, you need to divide the population so that the cells don't overgrow on each other and like compete for resources and just like grow out of the dish basically. And you could just move some of those to a new dish, but you only need like so many cells to work with, right? So you actually end up killing a lot more cells. They don't have nervous systems, so they can't feel the pain. So don't worry about that too much. But because we knew that we could